Thank you very much, Keith. So welcome back to the second part of the first session. Thank you for returning to the room. If there is anything that that I say that you don't understand, just raise your hand. I'll try to make it a little bit clearer. So this session will be on starting materials to ASMF. So it will cover general issues on active substances, since from the contents of the dossier, the applicable guidelines, and I'll make as much as possible reference to cases that we have seen and discussed in the centralized procedure. As it was mentioned before, and probably will be mentioned again, here is a summary of where you can find the applicable guidelines, legislations, and standards to be met. You can see that overall, this can be found in the European Commission, on the EMA website, on the ICH, and in the QM. On these links that you see on this on the slide, and all the links that you'll see throughout the presentations, you'll have a direct understanding of what are the dossier requirements, the data expected to be provided, the standards to be met, and as well the legal responsibilities from both active substance manufacturer and finished product manufacturer, and MAH. For example, for the active substance, one of the main legal responsibilities is the responsibility to communicate to the MAH or to the applicant any change to the manufacturing process or control of the active substance. Another legal responsibility of the active substance manufacturer will be the keep up to date with the state of the art. And an indirect responsibility would be the information to the competent health authorities through the MAH of any new information that can impact the benefit risk of the product. Now it's a simple question: what is a chemical entity? But it not always has a very easy answer. The definition of a chemical substance, or a well-defined entity, as is referred in the directive, can be achieved by exclusion to what is a biological and what is a herbal. So, like a biological, a chemical entity also needs to be also needs for its characterization and determination of its quality a combination of physical, chemical, and biological testing as adequate or as applicable, together with the production process and its control. The difference being, of course, the source of a biological substance is biological. On the link here, you will find a non-exhaustive list of non-recombinant biological sources, which has been agreed and has been up for some discussion in the past. With regards to herbal, the difference being the absence of a chemical synthetic step between what would be a chemical and what would be a herbal. Of course, the importance here is what guidance would apply, and what legal framework would apply to the requirements and expectations of the dossier. Okay. You've already seen this information before. These are the contents of the data supporting the quality and manufacturer control of the active substance. This is not in order, but it focuses on how important the control is in the whole quality data package of your active. In general, all the investigation, all the studies, all the research, all the discussions that you will have to provide in the different sections will stem ultimately how the substance is controlled, or how it will need to be controlled to be at an acceptable standard. When defining the active substance, there are different things that need to be considered. For example, the different polymorphic forms. That is your active substance subject of 2, 3 different polymorphic forms. Will these polymorphic forms impact on the bioavailability or the stability of the product? Can there be any interconversion of these forms during the stability or manufacturing process of the active substance or the finished product? These are the some aspects that you need to be very clear and justify very well in your data submission. I included in the second bullet point, the chiral substances, a link to specific criteria and specific requirements for this type of mixtures of two enantiomers. Another, on the third bullet point, we'll have the multi-component active substances. So these are complex active substances. One of the examples that were mentioned before were polymeric substances, where you might have more than one species there. Another example is, of course, the fermentation products. On these products, you might have to justify if the activity will come especially from one of the species, and what are the other species' contribution to the whole, to the overall safety and efficacy of the product. Of course, this then will relate again to the control of the different species, both in the active substance and the finished product. We have other relevant properties, such as the nanomaterials. Of course, these are very specific, and there's already a session in the afternoon about this type of products. Then mixtures. It's important to know that mixtures are allowed in very specific circumstances, overall when there are safety or stability issues with the active substance itself. It would not normally be acceptable for an active substance to be presented as a mixture of an active and an excipient. And this, of course, has an impact on when the manufacture of the finished product actually starts. So when a GMP certificate and a manufacturing authorization would need to be presented. The last point I would mention in this slide would be the expression of strength. We see a lot of, or some, applications where the entire development of the product was made based on the salt or the hydrate form, etc., of the finished product, of the active. This would mean that when the submission is made, you have the strength of the product as 20 milligrams, example. Then when you take up the salt part of the substance or the hydrate part of the substance, the strength goes down to a decimal case figure. This is often not desirable for the marketing application holder, but these are the rules. The expression of the strength has to be based on the INN for the purpose of the legislation, and on upcoming generic applications in 8-10 years' time. So when you are developing your product, consider starting your clinical trials already with an expression of the strength that would meet the criteria of the legislation, as well as your own interests and suitability for the patient. Now we go to another section of Module 3, the declaration of all manufacturing sites involved need to be declared. These have to be declared for in both the ASMF or the SEP, if that's the case of it, in the 32S and in the application form. So all this data has to be consistent. All sites have to be declared, and if you have a specific route of synthesis only perform at specific sites, or specific testing only perform at specific sites, you need to make it very clear in your data dossier. the active substance is broadly defined by its impurity profile. So if the use of these different synthetic routes and this manufacturing processes yields a substance with a significantly different impurity profile, this will be considered as two sources of API and not only one source. So you would be expected to provide the applicable data to differentiate between source 1 and source 2. I include again some links on process validation guidance. It's important to say that it's not expected to provide process validation data for active substance unless they are sterile substances. Okay. On this next slide is one of the hot topics on active substances in regards to starting materials. Currently, defining a starting material at an earlier stage of development can ensure that all necessary requirements are met at the time of submission of your marketing application. It can be an issue, and it can delay the approval or even reject your marketing authorization if you didn't do this work beforehand and you didn't correctly define the active substance, the starting material at the right stage of the synthetic process. This would, of course, lead to a lot of work during the assessment period. So during the clock ons and clock stops of your application procedure, and sometimes you cannot meet the data within the regulatory time frame. The choice of the starting material is driven by understanding of all critical steps of the manufacturing process that might impact the impurity profile of the active substance. The directive says that the applicant and the MAH holder has to be informed of all critical steps of its product. I'll have to restart that over again. The directive requires that all information necessary for the applicant and MAH to be held responsible for its product needs to be made available for the applicant. So it's on that basis that these critical steps, which are steps that can significantly impact on the impurity profile and therefore safety of the active substance and therefore the finished product, need to be included in the open part of the ASMF, so need to be available for the applicants. And this, of course, is where the bar will need to be put. Again, an adequate control of the specification of the starting material is not in itself sufficient to guarantee the quality of the active substance. As it was mentioned before, is a summary of the control and GMP compliance that will provide the necessary guarantees that your active substance meets the necessary criteria to be put in or to be acceptable in a marketing authorization. You will find more detailed information on the starting materials in ICHQ 11. I have included some overall bullet points on this 4 criteria. So it's defined chemical properties and structure, not a non-isolated intermediate. It's a significant structural fragment. It needs to have an adequate specifications. However, having a very tight and adequate specification does not in itself will make an intermediate an acceptable starting material, because there will be steps upstream that will need to be controlled and need to be available to the market, to the competent authorities. And MAH and needs to comply with GMP guidance or GMP standards. Going upstream will also mean that your QP declaration will need to be updated to include any sites that were not included initially, should the starting material need to be redefined. And I heard a lot of interest and a lot of understanding of what a QP declaration was, so you can understand how critical it is to start off at the right level. It cannot be stressed enough how important and how critical the definition of starting materials is in your data package. Here is a breakdown of the main discussions held by the QWP core team on scientific advices and marketing applications of applications for the last approximately 2 years, and you can see that the discussions on starting materials have represented more than half of the entire discussions. Now we move to another section of the content of your data package for active substances, which is the impurities. So I skipped one slide. So I'll go back again. On the manufacturing process development, you are expected to give a thorough analysis and discussion on all critical quality attributes of the active substance in the finished product. These are not only the quality attributes for the manufacturing process of the substance and the finished product itself, but also the in vivo aspects that can affect the performance of the product. The manufacturing changes during the pharmaceutical development and clinical trials need to be declared in the dossier. There was a question this morning about this. There isn't a general guidance on all specific changes that can be introduced, so it's up to you that has the better understanding of your product to provide a discussion on how a change in the synthetic process or a change in the conditions of that process could potentially require more in vivo and in vitro data to substantiate that the product that you used in your non-clinical and clinical studies is equivalent to the product that you are submitting for the market. Finally, the satisfactory control of the manufacturing process, particularly when end testing may not fully control all attributes of the active substance. This is very important with regards to those complex active substances where it's not always possible to have an analytical method that can fully identify all species or to identify the quantity of all species present in the finished product. This will be the example of Copaxone or of other fermentation products that have many multi-components or many sub-components. Finally, the slide I skipped, the impurities section of your dossier need to be quite exhaustive to all potential origins of contaminants of your active substance. This can go from the TSE, the vestigial DNA, the impurities of your reagents, the impurities of your solvents, all of those impurities of your intermediates. All of these aspects need to be discussed, and the absence or non-testing on the active substance itself need to be justified. I included, again, some links for ease of reference. Here are the reagents, residual solvents, process intermediates, metal catalysts, byproducts and carryover impurities from raw materials, degradation products. Of course, the amount of discussion will depend on the type of product. So it's not only how the manufacturing process of the active substance is actually happens, but is also what type of product do you have? Is it short-term, long-term? Or I will better phrase as a chronic use or acute use product is what type of indication it is, what type of administration routes it will have, and of course all of this will have impact on how much you have to address and how critical we will be on this side. I'll leave the genotoxic impurities discussion for the next panel this morning. Finally, on that very important part of the of your submission is the specifications and methods to control the active substance. These will be a summary of general parameters as well of specific. Again, these specifics are tied in to how your product is expected to be processed in the finished product, how stable the active substance is, how critical it is the particle size for the bioavailability of the active substance in the patient. All of these parameters that can be included or omitted from your specification, as well as their limits, will have to be justified based on development data, compendial standards, and of course the data that was or the product that was used for the non-clinical and clinical studies. The batch data should be at least on 3 consecutive batches and not less than 10% of the commercial scale. Any outlier or any deviation from guidance would need to be explained and justified. Providing this data will not only show that you comply with the guidance, you comply with the specifications that you are proposing, but it will also show how robust your manufacturing process is. Stability, you will have to comply with the ICH standards. These are available on the link below. It's important that you mention which batches are you using for those stability studies, if they are pilot scale, if they are commercial scale, if your container closure system changed between the studies that you performed and the products that you are proposed to put into the market, and whether any special labeling conditions apply. Now for more administrative but also important information is the submission of data. The data on the active substance can be submitted as an open file, meaning that the full information is in module 32S. It can be presented as an active substance master file or as a certificate of suitability, and Andrew McMath afterwards will provide more information on this. Of course, we have moved with time, and the submission in data in general went from paper to electronic, and now it's through e-submission gateway. The different national competent authorities have different requirements to the submission of data, so it's highly recommendable that you visit these standards so that you comply. If you don't meet these standards, it can be an invalidation issue, which can delay your the start of your marketing application. Again, the ASMF procedure its immediate aim is to protect all confidential information, protection of confidential intellectual property or know-how of the manufacturer of the active substance. Its scope is on well-defined actives, such as new substances, chemicals, herbals, substances that have a monograph and substances that don't have a monograph. These exclude biologicals, excipients, and finished products. While saying that the ASMF is there to protect the intellectual property of the manufacturer of the active substance, it's always important that the finished product and the applicant has enough information to be able to be held accountable for the quality of the product that is being put in the market. The contents of an ASMF is the same as the contents of an open file. Is divided into an applicant's parts, which also has to be copied in module 32S, and this is a quality assurance to be sure that both active substance manufacturer and applicant and finished product manufacturer are aware of the quality standards being applied, and the restricted parts that will only be available to competent authorities. Both these parts have to be submitted by the ASMF holder. In case more of one supplier of active substance is presented in the dossier, you'll have to submit a consolidated specification that will be valid for all sources of active substance that you are applying for. We strongly recommend that you visit all the available pre-submission guidance on the EMEA and PMDH websites. I have one minute, so I'll be as short as I can be. I take this opportunity to present you with the ASMF work sharing pilots that has started in December last year. It was recognized that the same ASMF is used in different products and across different procedures. This, of course, led to a duplicated assessment, diversion decisions, frequent ASMF updates, not only triggered by the ASMF holder, but also at the competent authorities' request, an overall reduced oversight of the ASMF, and an increased workload for all parties that was not desirable. A joint group with the PMDH, CMDV, the CHMP and CMDV, and ADQM as well, are starting on this ASMF work sharing pilot process. This is based on the current legislative framework, and the tools that we have to make this happen are the letter of access, the ASMF submission details form, the ASMF assessment report repository, which is only available to competent authorities, and the EU ASMF number that is allocated to your ASMF should you want to be part of this pilot. So I hope I met my, no, I still have one slide before my minute is over. There was no impact in procedure timings. It reduces the competent authorities' request for ASMF updates. It harmonizes ASMF assessments, reduces workload for all parties, so the benefit is both on you and on us, and improves oversight of an ASMF during its entire life cycle and its use in the different procedures, different products, different member states. So this was all. Thank you very much.